So the first thing I wanted to talk about is the mitochondrial import. But first, a break. The reason fatty acid degradation has made it into the mitochondrial metabolism is weak is because it occurs in the mitochondria, so remember that. And mitochondria is awesome because most of our energy production occurs in the mitochondria, and without it, we would be very sick. In fact, there's a whole slew of mitochondrial diseases where one of the most prevalent symptoms is muscle fatigue and weakness, because muscle is starved for ATP because their mitochondria don't work so well. All right, so carnitine transporter. Now, I've mentioned that just right now that mitochondria has two membranes, so two lipid bilayers. While when people talk about mitochondria in textbooks, they tend to only draw one bilayer. And that's because the outer membrane of the mitochondria has a bunch of pores inside, and so things can come in and out fairly easily. So for the trying to simplify concepts, artists tend to drop one of the mitochondrial membranes in a lot of illustrations, which is incredibly confusing for students. So acyl-CoA gets into the outer mitochondrial membrane using the VDAC porin channel, but it can't cross at all into the inner mitochondrial membrane. There are no pores on the inner mitochondrial membrane because its big job is to separate protons from the mitochondrial matrix, creating proton motive force, as we will learn when we talk about electron transport chain and ATP synthase. And while coenzyme A and acyl are small enough to fit through the VDAC pores, because they're big, the cell actually keeps a separate pool of cytoplasmic coenzyme A and mitochondrial coenzyme A, and those two never meet. So instead, a shuttle is used, and the shuttle's name is carnitine. Carnitine reacts to acyl-CoA, releasing the cytoplasmic CoA, which can then diffuse back through the VDAC pore back into cytoplasm, and making acyl-carnitine. And this reaction is catalyzed by carnitine acyltransferase 1, which is the one you find in the inner intermembrane space of the mitochondria. So this whole business about cytoplasmic side, not true. This is the intermembrane space. And then acylcarnitine can be recognized by this translocase enzyme, translocase protein, which is stuck in the inner membrane of mitochondria and can translocate acylcarnitine into the matrix of the mitochondria. There's a separate pool of available mitochondrial coenzyme A's, which promptly react with acylcarnitine, facilitated by carnitine acyltransferase 2 reaction to form free carnitine and acyl-CoA molecule, which is now inside the mitochondria and can be degraded. Once acyl-CoA is inside mitochondria, that is the only thing that will happen to them. It will be degraded. So, and then as the last step, carnitine will be exported back using the same translocate back into the inner mitochondrial membrane. And what I wanted to mention, I dropped that slide from the presentation, but carnitine is one of these wonder supplements that people who like to grow big muscles take. However, you can buy it in jars, like pretty much all the other prosthetic groups known otherwise as vitamins that are sold by health food stores. However, the idea that the carnitine actually does anything to help you metabolize fat is very shifty, because we get plenty of carnitine in our regular diet. We don't need more. All right, so what happens then to the fatty acids, currently known as acyl-CoAs, that have been transported into the mitochondrial matrix? They are degraded by beta oxidation. Now, beta oxidation is a very cyclical reaction because every time a beta oxidation step is complete, what you release is acetyl-CoA, which only has 2 carbons. And then you go back and clip off more carbons from the acyl-CoA chain. The process has 4 steps. The oxidation step, the hydration step, where water is added to destabilize the molecule, a second oxidation step, and the thiolysis step in order to release this CoA in order to attach a new acetyl-CoA molecule to the shortened acyl-CoA chain. I have another slide following this one where we're going to go through this in greater detail, but right now I wanted to discuss the enzymes and the enzyme names. What I want to point out here that there's 2 oxidation steps catalyzed by dehydrogenases, and these dehydrogenases, which are enzymes that catalyze redox reactions, in the first step convert FAD to FADH2, which can be used in electron transport chain. And in the third step, produce an NADH, which can also be used in the electron transport chain. So beta oxidation already produces energy or sheds electrons that can be used for production of ATP, even before this little acetyl-CoA is even fed into the citric acid cycle. The other 2 enzymes are a hydratase because it catalyzes addition of water, it hydrates to the NO-CoA intermediate here. And then the final step, because it involves sulfur, is a thiolase because it catalyzes thiolyses, where a thiol group is added to this particular location, and the previous coenzyme A is removed as acetyl-CoA and sent on its merry way. And I just want to stress one more time that if you have a redox reaction involving some sort of electron carrier like FAD or NAD+, the enzyme responsible for the job is a dehydrogenase. Okay, now let's look one more time at the cycles of each beta oxidation step. At each beta oxidation step. And in the previous slide, we looked at the 4 steps of each 4 reactions of each step. Now we will see how coenzyme A molecules come in and leave as acetyl-CoA. So you appreciate why it's called beta oxidation, because it occurs in the beta carbon, and the fact that with each step, a new coenzyme A comes in to grab onto the shortening acyl chain, and 2 carbons leave with each step. So we're starting with this palmitic acid here, palmitate CoA, and we have CoA attached. There's our sulfur and there's our oxygen. So the first step here, you can see that the pink coenzyme A with the alpha and beta carbon shell off right there. And during the reaction, a different colored CoA, in this case blue, came in, and in my diagram I colored it pink. So you can see that the orange CoA left, but the thiolase added a pink CoA at the very last step. Now the pink CoA left, but the thiolase added a green CoA in the very last step. And the release is the addition of a new CoA molecule and the release of the previous K, all is catalyzed during the thiolase step. The other 3 steps are preparation for the break. And finally, you can see that the blue CoA came in and the green CoA left. Now, when you have unsaturated fatty acids, things get somewhat more complicated. So before beta oxidation can proceed, things get stalled a little. So at first, when these 2 double bonds are far away, since all of the entire beta oxidation process is occurring right next to the CoA on the alpha and beta carbon, the reaction will proceed fine. until the double bond here is encountered, because the thiolase, the dehydrogenases cannot prep it for breakage. So instead, a different enzyme comes in. It's an isomerase, and its job is to move the double bond and make it make it converted from a cis conformation to a trans conformation. So you can see that the double bond is shifted from the being between the third and fourth carbon to being between a second and third carbon right there. Then the result of it is that then a hydration reaction occurs where more hydrogen are added, and FAD comes in, which harvests the electrons, converting it back to a double bond, to a trans double bond between the second and third carbon. Then another reductase comes here, and the reason, and even though you see an ADPH being involved, you can see that it's called not a dehydrogenase but a reductase, because it actually donates the electron and leaves electron empty. So it's a reduction reaction as opposed to an oxidation reaction. Then the isomerase can come in yet again for the second bond, and so forth, allowing the isomerization, combining isomerization and a reduction to convert the unsaturated fatty acid into something beta oxidation can actually handle and break, because this now looks like the trans-enol-CoA that is capable to be hydrated and then oxidized during the normal beta oxidation reaction. All right, so pretty straightforward, and obviously when obviously because beta oxidation occurs at the level of 2 carbons leave per each cycle of beta oxidation, we can convert an 18 carbon fatty acid into 9 acetyl-CoAs. And if it has unsaturated bonds, slightly fewer CoAs are made, but that's okay. So in a way, unsaturated fatty acids produce slightly less ATP than saturated fatty acids, but they're still critically important dietarily, because we like our unsaturated fatty acids for a bunch of synthesis processes.